Well, hello everybody. Thank you for coming to the United Diversity Metabolic Focus session. I'm very happy to share the session with Yulia today. So my name is Boxiang. I'm a PI at the National University of Singapore. My lab focuses on alternative splicing and also metabolic diseases. And Yulia, do you want to introduce yourself?
Thank you, Boxiang. I'm Medvedeva, and I'm a group leader also center of biotechnology in the Technology Research Center, where this project single cell analysis for type 2 type 2 diabetes in addition to medication. Um,
Those of you, for many of you, you've been to these sessions before, but in case for the first time to HCA or you missed the breakout session yesterday, I'll just briefly talk about the logistics of today's breakout session. So we'll start with introduction of metabolic diseases and diversity, and then we'll give out the microphones to people for the discussion session. So there will be one person handing out the microphone, so please raise your hand if you want to speak. And we do encourage you to as much as possible. The purpose of today's discussion session is more on the community engagement. And also there will be two scribers to take down notes, and the notes will be shared with you after the session. Okay? And please feel free to interrupt me if you have any questions. So just to sort of break us in for today's topic. So this session is called Metabolic disease, and involves many types of, especially in developed countries, right? So these diseases will be obesity, type 2 diabetes, like this, and also involve cardiometabolic diseases, for example, CAD, and for example, hypertension and hyperlipidemia. Genetic diversity actually plays a key role in metabolic diseases. And plays a role in many diseases in general, but especially in metabolic diseases. So a few examples, right? For obesity, the destination for obesity BMI threshold is different each group. So, there has been evidence that Asian has a higher likelihood of getting type 2 diabetes with the same BMI. So there's a range of evidence pointing that there's genetic influences on disease susceptibility. So today's discussion, I think we will revolve around these topics. One is, we have been sampling blood quite a bit, and we see the session more than yesterday. But for metabolic diseases, there is inflammation component. There is chronic inflammation, for example, both rating chronic inflammation for type 2 diabetes. But are there other tissues that we could sample beyond blood? I know many of you in this room is working on a diverse tissue types beyond blood. What are the next steps, and what are the available tissues which we focus on? And also, we want to explore the soft tissue communication, right? We know that there is certain relationship, for example, between your gland and your brain, just when you simulate your appetite and diet. So how do we not only connect the cell types within the same tissue, but connect on the tissue level? We know that environment is a key role in determining a wide range of diseases. And this is especially true for metabolic disease, because we know that diet and exercise are the most key determinant of whether a person gets diabetes or type 2 diabetes or not. Last but not least, we want to connect genetic regulation into how the susceptibility of metabolic diseases, right? And this would also involve, how does genetics play a role in explaining people's ancestral groups' susceptibility to diseases. Okay.
Since I'm more from investigational side rather than biology, so I just I think that if the metabolic disease, which is a really a lot of components, it's important to so to figure out research how we can benefit from the metabolic late in during their human research.
We'll try to speak less and we open the floor for any comments. So perhaps we could start with question number one. What are the key cell types and tissue types driving metabolic diseases across a wide range of metabolic diseases, and what tissue types we should focus on? Just in case you cannot see them, blow it up. And also, I should say that there are funders in the room, so our discussion would also help them drive potentially drive them different directions for future directions for future people. So, frequently, frequently, hey, yeah. Very bad. They're so red.
可以可以，可是他们6点半才出来，他的冰会融吗？呃，可以可以可以，还是有盐还是什么，可以久一点。
with COVID-19
哪里？
within different population.
对，
even though
可是他们他们拿走了吗？还是还在？
have
OK，他们应该开始清理了，但是
We could organize the infectious disease
Uh-huh.
oriented bio-network. And
可以可以，谢谢。
we have many different
Bye-bye.
types of the infectious agents. Like some people can think focus on bacteria, some
Do you think it's possible that for samples of tissues which are really difficult to collect, we can find some blueprints of their condition in some other cells or tissues which are much easier to collect?
Brain that so the REM maybe there are some people
Okay.
get brain specifically for the obese. Here, like for example, Rosneft, they focus on AD. There's many brain collections for neurodegenerative diseases. But I think that you mentioned it's very rare to collect brains for non-neurodegenerative diseases, and also especially for obesity that for non-elderly people that even run. Any other systems that people
Before we decide which tissue that is feasible to collect it, I think very that either these like because that figure these. I think I'm going to before that this the algorithm and what is it that half of trade that is different conditions of people, right? So that also and the consequences of different issues relevant to the I don't know, it's very interesting. Describe the place arrived and maybe actually ancestry and ancestry which particular things like that. Last year and also from but life with other ancestry and with people. For example, a paper they discovered first they found out that the Indian Bangladesh which is a relatively young and but it also like that. Using sample, we want that other roughly this Use the laptop. Clearly,
So Michaela is leading the cardiovascular biomarker, and I have a quick question for you. Do you have a wish list of what is the sample you're currently collecting, and what are the sample collections? Hi, I'm also working on the production of the food and it's, I prefer the thought and discussion of metabolic question rather question of what and how we set selection of those of the need to do this analysis. Because probably most of the issues that are yeah. And so I'm wondering, like first people blood brain, between liver, like there's so many issues that are like very relevant in cancer, and whether rather on the metabolic side, how to show that to be able to do analysis throughout, metabolic issues, and whether like, things that would about that. That's a very good question.
Wearable data is what. So continuous glucose monitoring while they're not. A lot of wearable information provides that next.
Quick follow-up for both of you. Of course, wearable is a very nice source of metadata. Are there any other metadata that you think is important? But I guess also clinical data would be very relevant. People have other clinical things, right?
Yeah, hi, hello. I'm Maritze, I'm coming from Qatar. We are trying to immunoprofile the immune cell, all the immune cells in adipose tissue. We are working in subcutaneous, these are adipose tissue because they are big difference, but also what we are facing is like, first we start focusing on all the Qatari females because they have this obesity. So our goal was to compare healthy obese and metabolic disease. But our core from Qatar, we are ranging girls that go to bariatric surgery from 20, 30 years old in this range. And we wanted to compare this core with a Spanish core in order to see, like genetic difference and everything. But then we realized all the samples we got from Barcelona, the range of age is completely different. And also, I think this is very important because a 20-year-old person that has been obese, the lipotoxicity, the immune regulation is shorter than someone is. So, I mean, we are like in the same disease, but completely. I don't know, it's getting that this data, and I think for that, this metadata is very important also in the time frame of the disease that we are capturing, because maybe what we think is a healthy obese, in 10 years will be a type 2 diabetic or not. So this I think is very important in order to map properly. I think the clinical metadata, the metadata in general is just been a problem in handling the athletes. Yeah, you have to run up the people and say, please, can you fill in these tables? Some people have collected results. So I wonder again if we can, in this case, which is metabolic disease, it is somewhat easy to have a group of people sitting down and making a list of minimal information that are needed. Then somewhat circulated to the community, have a consensus or a discussion of the pages. If you want to contribute, you can please not feel you collect these data. Then it is somewhat be able to communicate to people that are doing the studies of planning to do it. You think ahead and try to collect the data. This is collecting the market. It's the mission. Yes. It's not that way.
Please go ahead. My name is Musa Khanna. I also the team that in the introduce the human cell aspect female placenta from African women. Now, I was wondering why this is not a complete abortion, and the process looks like it has endocrine function during pregnancy. And of course, if we look at the pH complexity, which means that people are running because of the pregnancy effect on metabolic function during self-movement and other things. When wouldn't we include it in certain organ or tissue that metabolic function? We have collected the 20 gene therapies and our lab has been listening at questions around visiting the question to yeah, I think that would be a very good criticism to that. The vaccine suggestions and which is true that if you have, it's important to have community opinion and also the contributions of physicians to practice them. So it sounds like you already have some persons at bank, and it's very important for example CDM and other metabolic related diseases in culture. So I think that's a very important decision to put a note of.
Also, if I wanted to add to the discussion having this speech and the different also the difference between alpha and beta. I think to do this kind of analysis, yes, we need to collect the different tissues from different parts of the body because of abdominal fat, it's not the same. And probably it goes to the muscles now because different muscles will differ. And this is also important. And it's so easier to collect, for example, tissues from different parts of the body, some same individuals, but continues.
Yeah, I want to respond to you. Comment officer. I'm Branko. I'm responsible for the data integration for the adipose tissue network. So it's, I guess, very relevant here. We actually have multiple like data from depots. So we are currently collecting all the data, and then we'll see what we actually have. This metadata is also something that we already discussed. We have a quite long list of patient-associated metadata that we are planning to collect. And I plan on also, like, based on this discussion, maybe to discuss with the BioNetwork, if we can kind of organize this human cell atlas wide, then maybe some people can use this list as well. I guess that's very relevant then.
It's just a quick shout out to the adipose cell website. So they recently had a white paper published on Nature Metabolism. I encourage everyone to check it out. And there was a call to action on what are the adipose defaults to collect. So for you, do you mind telling us, I know people can read the paper, but is there any sort of wish list of adipose default that you want to collect as prolactin samples?
Yes, so the white paper is not my paper, but it is associated with our lab. Currently, we have 4 major depots, I would say, that is in the neck. So we have subcutaneous neck and deep neck. Deep neck is associated with brown adipose tissue, which is very interesting to look at as well. And then, of course, we have subcutaneous abdominal and visceral, and then that is still quite broad. So we have actually data from multiple regions in visceral, like around the organs. There is I'm discussing now with from some group leaders. They have, I think, 33 locations in the body. So I think we're covering a lot now. I don't think we have, for example, adipose tissue from the heart, so epicardial. Also within the bone marrow, but I mean, adipose tissue is so spread out over the body, so it's actually quite difficult to then collect everything and then make a meaningful analysis. And so it's just a proof. We just have to progress and see what we have in the end.
I'm cardiac surgeon from South Korea, and we are also collecting many tissues, like including valves and like great vessels like aorta and also heart, and also some of the epicardial tissues. And we have collected more than like 800 tissues from like patients who are undergoing surgery, and they like as like it's like related to the like aortic stenosis or the atherosclerosis. Like most of the patients, they have like hypertension or like diabetes and also like obesity. So I think like we didn't do any like subgroup analysis or something, but it will be really wonderful if we can see like how the tissues are different, like regarding the comorbidities of the patient. And also like he also mentioned about the epicardial cardiac fat. And I think that's also like very important tissue currently for in like various situations. And I think like mental fat can be also good source of the adipose tissue.
So I guess a quick summary for question number one. Thank you very much for contributing these ideas. So what I have noticed, and also what they're described in those afterwards, is that adipose tissues across various parts of the human body, and also we're lacking some, for example, the epicardium and adipose tissue which we don't have, and also the heart and arteries across various parts of the body. Someone mentioned placenta, which is important for childhood, and also mother the GDM. And I think I missed some. Yes, so the hormonal gland, thyroid gland, and adrenal gland, and also of course the brain, actually very nicely put. So it's I guess there's a few follow-up questions that I want to ask you, right? So with collectively in the future, I think that when we walk out of this room, we want to have like a rough idea of where we want to be. There's three key decisions that we have to make, right? If we have to prioritize certain tissues, and if we have to prioritize certain diseases, what diseases and tissues should we immediately focus on at the next stage of the HCA? So the current stage of the HCA, as we well aware, is focusing on the healthy tissues. And the phase 2 of HCA will branch into diseases. So if we want to focus on certain diseases and certain tissues, which diseases and which tissues we focus on the field? So that is also needs to be remarked for the editors. We actually have more of these. Those are going to be individual. We actually have a lack with a lean individual, and that's more difficult to select them. So that's also kind of a call out to people that have maybe lean based kind of to keeping that. I think we are mostly interested in the fact that we might like one of the diseases that we are totally focused on activity, and that's nice.
So I'm curious, listening to the conversation, it sounds more like metadata seems to be the priority rather than the tissue itself, because these diseases are all systemic. So I wonder, within the current data sets that are available in HCA, if anyone has looked across tissues, if at BMI or some other variable to see if there are signatures across tissue types, or if some other signature that can be picked out within that may give some direction about specific tissue types or other areas to look at, rather than focusing on a specific tissue, focusing rather on the marker or the metadata or the condition that is occurring in kidney, in immune, in adipose, and see if there's something there to help guide. Has anyone looked across? Can that be done? Is there the right metadata? So I know the answer to this already. BMI is not present across all tissues, but is there sufficient metadata of a large portion of the corpus that might help to guide some of that thing?
Just to respond to that, I believe that the banks that is still we see BDP and what for them announced that does have been done. I think that adequacy is the first experiment in that we can close out different. Yeah, so we will see what that yields and go forward and insight and so these kind of questions. I think it's going to be difficult to find one tomorrow to connect. But it's definitely something to think about. Yeah. So just on the metadata front, I think so for us it has been actually quite difficult in terms of like because when we publish a paper or when we publish a data set, what we do is we basically go through it, maybe focus on healthy ones, we select the ones that have very relatively healthy BMI and no metabolic disease, no aspects of that. But this requires us to actually connect to the hospital records. And this is something that I think I want to bring up here. Like most of the biobank, for instance, we actually have like extensive aspects of electronic clinical records. And I think this is something that we can link if this is possible. I know this varies from country to country. I think this is something it would require someone from higher up to say coordinate this effort. Say now we collect this data to say a kind of like a set of rules or regulations that we can actually access certain clinical records from those samples, because everyone here in the room have different interests. They would they would focus on different phenotypes or different metrics. And I think taking access, allow access to the actual primary and secondary clinical record would be extremely useful in these cases. I just want to sort of quickly summarize the points raised by the audience. It seems like the metadata is quite important. If we want to leverage the existing data to analyze the different disease states. But here's a follow-up question for the people in the room. Do we have a minimal list of metadata that we need to perform these analyses using different data, right? I think a few has been mentioned, for example, BMI, sex. I think the list of metadata is very, very, very long. But is there minimum essential list that we can collect and agree on that we can send out for people to collect? And hopefully without much needed funding to collect these metadata. The age and sex, and also ethnicity, and also I think so. Now we are working towards disease samples. I think it's also very useful to actually, when we run the analysis, we have to pair that with some sort of like genotyping or DNA sequencing platform, so that we can actually now then in a statistical genetics world, and we do a reward, we will usually correct traits for in the regression model to be insert the first genetic species, 10 genetic species in the model. I think this is some sort of the things that we should insert. So all the genotyping data should be paired with that, and then with that we can then stratify our data into different ethnicities that we. Then we need to some comments because when we collect the data from we had discussions about the one all the organs, a lot of people and then in the biomarkers, I think I'm not 100% sure, but I'm almost sure. Let's say this is basically all biomarkers always happen in life. I'm not 100% sure, but I think a lot of biomarkers there's a basic common, sex, then what's going to have for the system. And for the different tracking data sets within biomarkers so that does about obviously humans will have to repeat that's what I mean. But to be honest, my question is really whether this mechanism is important after the strategy going for the mental giving factors or something. I don't know. Then I don't know. It's just kind of, yeah. Yeah.
So we have seen over 2017 cardio data at presentation. Normal analysis is the challenge with metabolic disease is that compared to cancer and many other things well, that whether people are on statins and not on statins, whether they exercise, or they don't exercise, all those environmental factors, whether they're in the region of disease, whether they're not exposed to disease or not, they all leave a significant and the molecular. And I think that's the most scary about the whole thing. Is agents that, while it's important, it's just it's not where it's not enough. Because if you want to really unpack the question you are asking, otherwise we're just in front of this or something like that. Where it's older drug, yeah.
With I think for that, we can say, like yes, exercise. How do you quantify exercise?
Sorry, I think this is something I continue to work going forward. It's just to understand the mechanisms, and just a matter whether cardiovascular hypertension, diabetes, I think they all have the same pattern, and there's so much comorbidity between them. Is that the design has to be a little bit different to other diseases such as infectious disease. I think that's very unique.
I was also thinking about the systematic biases that are involved. So, for example, for nutrition, people tend to underreport the amount of calories they consume, and I'm guessing a lot of this metadata is self-reported. So I'm curious how significant do you think these biases are, and how would you go about reducing them?
It's a 20 cases way to say whether people misrepresent or misremember their practical service.
I think both of you raised a very important question. So of course, like statin, but perhaps metformin, those are the very important drugs. Exercise and diet. So for drugs, they usually report fairly accurately, up to compliance. But then for that, I totally agree with you that people misrepresent themselves. They're more usually the amount of exercise they do. I'm not sure if there's like a standard in collecting these data. Is it using the wearables or because this, in a random controlled trial, we would use these filters to do that, but here it's self-reported.
So for people, for bias, for example, each country has their own 144 a list of foods, for example. Middle East have their own, and Indonesia have their own. Singaporeans also have their own, and the Western have their own. So what if you cannot select the raw data, but then we will have buyers and founders, and you select only the depending now, and that was whatever, like you know, because there are different calculations for each item of the food. So it's complicated, but it's also that's why the methodology is one of the most complicated one. So then you have so many buyers, and it's much easier to generalize everything. The initial just measures metabolic in the blood, several of them that. For example, that's why I think you have first decide which metabolic disease that you want to look at. I don't say that all metabolic disease has similar. That's not true. That's different pathomechanisms, right? And they affect different. The effect of tissues are also different in different tissues. So first of all, we have to focus. For example, if you want to focus on diabetes, then we encounter a young diabetes, young onset of diabetes, and then just the stational diabetes, etc. But then you can all have a standardized blood glucose measurement or HbA1c. You decide with this that type of diabetes. But not all metabolism is I think. From all the different diseases, like let's take the three big, right? So H1C we measured. We also measured a few key things. So there is a kind of criteria for metabolic syndrome, for example. Five criteria. This is also very, very diverse because you have 5 criteria. You have a positive things to be that in the metabolic disease, that's the syndrome. But that's not if you look deeper into the muscular mechanism, because all of the like you need to have a high not BMI, but in ratios in birth. Female and male has different cutoff. Asians and Caucasian has different cutoff. Now, Middle East also has a different cutoff. The BMI does not represent the overall adipose distribution. Fetal fat is more relevant because they are relevant to induce disease. Then after race and concern, we will have a higher cutoff of blood pressure, and then triglycerides, and then cholesterol, etc. HCA, metabolic. So it's not easy. If you want to have a generalized.
No, it's not generalized. But so what's very interesting is supposedly, I don't know different countries. So I can only speak for Australia. If we go for like a 49-year-old, 50-year-old checkup, there's a standard blood report. In the standard blood report, certain blood it's measured, right? And so I was wondering whether that kind of measurement could be used. But it will be interesting to obviously in the committee to see, what is the standard healthcare blood panel that is typically done in the different countries, right? And I think that is a start, because then you are not asking for more collection. It's your standard blood work. It's already like the standard blood work that the country does and what is common between the different countries.
Okay, yeah, please. Yeah, so I have a standardized list here of all the blood work that we're collecting for the adipose tissue. So it is a quite long list actually. I don't know if it's necessary to discuss the metadata now. Maybe there is a way that I can kind of get it out. And then people can kind of just go through it and maybe comment. because we have like 80 values that people need to collect, which is quite a lot. And yeah, any suggestions are welcome, of course.
I'm going to give you one more question. I'm going to use my moderator privilege to summarize. Okay, so please go ahead.
Just a quick comment. I've been listening to all of the discussion that's going on. Everything is extremely relevant. Single cell studies are very expensive. So what we probably will need to do is to gather a cohort of individuals, measure all of these variables, and perhaps look at some extremes, let's say top 5% tail or bottom 5% tail, and contrast those, for which we will do the single cell study. So the study design needs to be appropriately thought based on these various metadata, lipid variables, BMI, HbA1c, etc. So we need to come up with a method by which we can plot the distribution. This is, of course, a multivariate distribution that we're plotting, and look at, top 5% tail versus the bottom 5% tail, and cell studies there. Let's learn about the extremes, and then we can think about the middle. Otherwise, who is going to fund hundreds of people? Or even though we've collected data on 1,000, metadata on 1,000 variables. So I think we need to combine practicality with the science.
So that's a very important point. About that. I think many people in the audience mentioned we have to combine practicality, which is the comprehensiveness of the metadata, right? So I'm not sure if there's has been an effort to systematically meta-analyze the data that's already deposited on field or cell by gene to see what are the most common metadata that's been deposited. Right? So, for example, perhaps every study would potentially have sex and age and maybe ancestry. I'm not sure. So there's different levels of metadata, as one of the audience has mentioned. Perhaps, especially within the metabolic diseases, where BMI, wasted ratio, and statin and metformin use is so important, perhaps there needs to be a specialized set of metadata, and we can have, for example, level 1 would be the same across all the biometrics, but perhaps level 2 can be somewhat specific to metabolic diseases. So the gentleman there mentioned that there's like a list of 80 different metadata that might be available. Perhaps we can, that's a list that we can work from, and hopefully not to expand on, but to reduce that list to a minimum list that everyone has the power to collect, because not every country use a comprehensive blood panel as you mentioned, and then we have to find the commonalities across the different countries and the countries. So I originally thought that we stopped at 5:25, but we started late 15 minutes, so we started at 5:40. So we can, in theory, stop at 5:40. So the session is 1 hour. I want to, sort of brainstorm with you, as your last exercise, collectively as a community. What kind of question, like if we want to start a project and push the funders for our project, what projects would you propose?
I have a small observation and comment to make. So one thing that I'm guessing is pretty obvious by now is that it's difficult to collect metadata that answers all our questions for all the kinds of samples that we wish to collect. But one thing that seems rather accessible is perhaps blood blood collection. So would it be possible to studies that do more in-depth analysis of the various interventions they are looking at, or various facts they're looking at, like environmental exposure, diet, exercise, on people who where these things can be readily quantified and controlled for? You draw the blood from them, and then you use blood as a kind of linking factor to be able to input missing metadata between samples. So I'm guessing that basically using blood as a linkage to kind of fill that in the knowledge as a sort of like integrator for these various issues and factors that you're interested in.
I think that works for live biopsies, but not so much for postmortem tissue.
I think what we would we sort of can look at the statistics. For example, national statistics from common diseases. For example, like in disease, in Russia, we know that in certain regions we have very low number of cases, and in some regions we have very high cases. So I think it's logical to compare with why we have so many cases this year, and it may be and lifestyle choices for the this kind of process can easily get funded because it's clear structure and we can at least something to do output.
Ideas, perhaps from the audience who have spoken before. Especially trainees, we encourage wild ideas, not just come to eyes.
Hello, this is Li Jiangfei from Cambridge Stem Cell Institute. So I'm wondering, when we collect the single cell data of this metabolic tissues that we propose today, how could we use this data? So one question in my mind is that how can we use this single cell data to help develop effective preclinical models for cardiometabolic disease? So this is kind of a future of our project. Yeah.
Just to clarify, your project is to use the transcriptomes and the functional genomics to predict the clinical outcome of the vaccine?
No, I'm just thinking about when we collect all the metadata and the single cell data of the tissues that we propose today, how can we use this data to do this that?
So I think one thing that we can talk about is to use the genetics, which can really find a target. But I see a hand raised.
Number one, diabetes is cancer. Second is diabetes. So I think diabetes is the most important disease group. There has to be a lot of things that there has a lot of questions. So I don't know whether it is really just like I said, and this stuff.
I think the nice thing about diabetes is that it's not an endpoint. It's actually chronic inflammation that influences all schools of other diseases, right? So So we can use diabetes as a risk factor to see how that changes different systems. I think that would be a very nice study to do across the entire HCA deposited tissues. You know, if we can see the diabetes metadata and how does that change the expression patterns across tissues. Okay, so we're right on the clock, 5:40. Thank you so much for your participation, and we'll share the scribe notes afterwards, and we'll perhaps start a discussion about what metadata to be collected and also how to analyze the metadata that's existing within the HCA. Thank you so much.